Okay, think about this for a second. Just, tapping your fingers or wiggling your toes. Seems simple, right?
Effortless, usually.
Exactly. But it's actually this incredibly complex dance happening inside your brain. Billions of signals firing just right.
Hmm.
But for more than 8 million people around the world, that smooth coordination, it breaks down. We're talking about Parkinson's disease.
Yeah, a truly devastating condition. You see tremors, stiffness, real problems with movement and balance.
And we've known for ages sort of the basic problem, specific brain cells, the ones that make dopamine. They start dying off. Dopamine's crucial for movement.
Right, the dopamine producers in a specific area.
But the big mystery, the real head scratcher, has always been why? Why do these cells in this place just collapse?
Yeah, what triggers it?
Well, today we're doing a deep dive into some really groundbreaking research, a new study from the Gladstone Institutes. And it points to a culprit that's, well, almost intuitive when you hear it. Overworked brain cells. Literally, cells working too hard.
Burnout at the cellular level.
Kind of, yeah. So our mission today is to unpack this. How could being too active actually lead to these neurons dying in Parkinson's? This is more than just cool science. It might really shift how we understand the disease and maybe even how we treat it.
What's fascinating here and what really grabbed me about this study is how it challenges some of the existing ideas about Parkinson's causes.
How so?
Well, the core finding, like you said, is stark. These specific dopamine neurons, when they're pushed into constant high gear, they don't just get tired. They seem to break down, their connections wither, and then the cells themselves die.
Wow.
And understanding this isn't just academic, for you listening, grasping why this matters could really change the game for future treatments. It's about the mechanism.
Right, the mechanism. Because that
Right.
why has been the puzzle. We know it's dopamine neurons, mainly in the substantia nigra part of the brain, the movement control center.
Hmm.
And we know they die as Parkinson's gets worse. But the weird thing is their activity often seems to increase before and, even during the degeneration process.
Yeah, it's been observed.
So the big question was, is that increased activity driving the death? Is it the cause? Or is it just the cells desperately trying to compensate a response to something else going wrong? Cause or effect?
Exactly that question. And that's what this Gladstone Institutes team, led by Ken Nakamura, with Katerina Rademacher as the first author, set out to answer. They came up with a really clever way to test it.
Okay, how'd they do it?
So they worked with mice. They used genetic tools to introduce a specific type of receptor into just these dopamine neurons. Think of it like installing a special on switch only in those cells.
Ah, okay, a targeted switch.
Precisely. Then they could flip that switch using a drug called cyanoclozapine N-oxide. But here's the really crucial part.
Yeah.
How they gave the CNO
How?
through the animal's drinking water.
Continuously.
So not just a one-time shot.
Exactly. That's the breakthrough element.
Yeah.
Previous studies might have used injections, giving these short, temporary bursts of activity.
Like revving an engine, then letting it idle. Right. But Parkinson's isn't a temporary burst, is it? It's a chronic, ongoing process. So this continuous CNO in the water meant the neurons were chronically overstimulated, day in, day out.
Okay, so they were mimicking that potential long-term stress.
That's the idea. It let them see what happens when these cells are forced to work overtime for an extended period, which is likely much closer to what might be happening, over years in the human brain.
So they set up this chronic overdrive. What happened? What did they see in the mice?
Well, the effects unfolded pretty quickly, actually. Within just a few days, they noticed the mice's normal activity patterns, their sleep-wake cycles, basically were disrupted.
Okay, so behavior changes first.
Then, after about a week, they could actually see physical changes under the microscope. The long arms, the axons that extend out from some of these dopamine neurons, they started to degenerate, to wither.
This is 1 week. Wow.
And then, the really critical finding. By 1 month, the cell bodies themselves, the main part of the neuron, began to die off.
A clear progression. Disruption, degeneration, death.
Yes. But here's the part that makes you really sit up and take notice, the connection to human Parkinson's.
Okay.
This damage, this cell death, it wasn't happening randomly all over the brain. It was highly selective. It
Right.
primarily hit the dopamine neurons in the substantia nigra.
The movement control area.
Exactly. And just as importantly, it spared similar dopamine neurons located in other brain regions, like the ones involved more in motivation or reward.
Wait, so it targeted the exact same cells in the exact same area that are affected in human Parkinson's?
Precisely. That specificity is incredibly powerful. It strongly suggests that their model, this chronic overactivation model, is tapping into a fundamental process that's actually relevant to the human disease.
It's like they recreated the specific vulnerability we see in people. That's huge.
It really is. It points towards overactivity being potentially a core part of the why.
Yeah.
So then they dug deeper, looking at the molecular level. What's going on inside these overworked cells?
Well, right. What makes them break down?
They found significant changes. For one, levels of calcium inside the neurons were altered. Calcium is like a crucial messenger molecule in cells, so messing with its balance can cause big problems.
Okay, calcium levels. What else?
They also saw changes in gene expression, which genes were turned on or off. Specifically, genes related to making and handling dopamine itself. Dopamine metabolism.
Ah, so the cell's machinery for its main job was affected.
Exactly. And this led Katerina Rademacher, the lead author, to propose a really interesting hypothesis.
Yeah.
She suggested that maybe, just maybe, when these neurons are constantly firing, they actually try to reduce how much dopamine they produce.
Reduce it. But isn't lack of dopamine the problem in Parkinson's?
It is, eventually. But her idea is maybe too much dopamine, or the byproducts of making it constantly, can actually be toxic to the cell itself. So the cell, in a way, tries to protect itself from this overactivity stress by throttling back dopamine production.
Like, okay, this pace is killing me. I need to slow down production.
Sort of. But the tragic irony, the hypothesis goes, is that this self-preservation attempt might contribute to the cell's eventual demise. It's struggling, it's under stress, it reduces its function, and over time, this struggle itself leads to burnout and death.
And that reduced dopamine then makes the movement symptoms worse.
Worsening the symptoms, which, as we'll discuss, might even put more pressure on the remaining cells. It seems like it could become this terrible feedback loop.
Wow. Okay, so that's the theory based on the mice. But how does this connect back to actual human patients? Did they find any evidence there?
They did, and this is another really compelling piece of the puzzle. They looked at gene expression patterns in brain tissue samples from people who had died with early-stage Parkinson's disease.
Okay, comparing the mouse findings to human brain tissue.
Exactly. And they found remarkable parallels. Those same types of genes, the ones involved in dopamine metabolism, calcium regulation, handling cellular stress, they were also turned down, showing decreased activity in the human Parkinson's samples.
The same pattern.
The same kind of pattern. It's like finding the same molecular signature in both the experimental model and the human disease. It really strengthens the case that this overactivity mechanism isn't just a mouse thing, but reflects something real happening in people.
That's incredible. Finding that molecular fingerprint in both places. It really ties it together.
It does. It makes the findings much more robust. Now, it's important to be clear, this study is brilliant at showing how neurons might die once they become overactive.
Right.
But it doesn't fully answer the step before that. Why do they become overactive in the first place? That initial trigger is still under investigation.
So what kicks off the whole process?
Dr. Nakamura, the lead investigator, suggests it's likely a mix of things, probably genetic factors that make certain neurons more vulnerable, combined with potentially environmental influences over a lifetime.
The usual suspects in complex diseases, genetics, and environment.
Pretty much. But he also brought up that vicious cycle idea we touched on.
Explain that again.
Okay, so imagine some initial factor, genes, environment, whatever, causes some dopamine neurons to become overactive and maybe start to fail or reduce their dopamine output.
Right.
This initial drop in dopamine starts causing subtle movement problems. Now, the brain tries to compensate. The remaining, still healthy dopamine neurons might get signals saying, hey, we need more dopamine output. So they start working harder.
Picking up the slack.
Exactly. But as this study shows, working harder, being chronically overactive, is precisely what puts them at risk. So the compensation effort itself drives more cells towards exhaustion and death.
Which means less dopamine, worse symptoms.
Which puts even more pressure on the next set of remaining cells. It becomes a downward spiral. The overactivity fuels the degeneration.
Wow, that's a chilling thought. A self-perpetuating cycle of burnout.
It is. And understanding that cycle is absolutely crucial if we want to find ways to intervene early. We need to break the cycle before too many cells are lost.
Which brings us to the hope, right? The potential implications for treatment. If overactivity is a key driver,
then the possibility, the really exciting possibility Nakamura mentioned, is that maybe we can protect these neurons by, well, by helping them chill out a bit.
adjusting their activity levels.
Exactly. Finding ways to modulate their activity patterns, to prevent them from going into that chronic overdrive state. If we could do that, maybe we could protect them from burning out.
How might we even do that?
Well, potentially through new drugs designed specifically to fine-tune neuronal firing rates, or perhaps by refining existing techniques like deep brain stimulation, DBS.
DBS is already used in Parkinson's, isn't it?
It is, often for symptom management later on. But maybe understanding this mechanism could lead to using DBS or similar approaches in new ways earlier on, specifically to normalize activity and protect the cells themselves.
Imagine that, slowing down the disease just by adjusting the workload of these critical cells.
It opens up a whole new therapeutic angle. It's not just about replacing dopamine anymore, but about preserving the cells that make it. It's a genuinely hopeful direction for research.
Definitely. So, wrapping up this deep dive, the big takeaway is pretty profound, isn't it? Parkinson's, this disease that steals movement, might be partly driven by the very cells responsible for movement, literally working themselves to death.
That seems to be the core insight, yeah. And seeing that process mirrored so clearly from the mouse model to the human brain data gives it real weight.
It really shifts the focus. It's not just that cells die, but uncovering this potential how and why this mechanism of overactivity leading to burnout. So for you listening, what really stands out about this idea, these overworked neurons?
And maybe here's a final thought to chew on, building on that. If this kind of cellular overactivity is a factor in a specific disease like Parkinson's, well, it raises a broader question, doesn't it?
How so?
Think about the demands we place on our systems generally, our daily activities, chronic stress, constant stimulation. How might these things, even without a direct link to Parkinson's, subtly influence the cellular environment?
You mean like contributing to general cellular stress or exhaustion?
Possibly. Could the way we live impact whether our cells, especially neurons, operate in a sustainable way or are pushed towards a state of exhaustion? What can we learn from this Parkinson's research about adjusting, maintaining balance and resilience at a cellular level for overall brain health? Something to consider.